Hello, everyone, and thank you for joining us today. I'm Karina from SynBio.Oxford. We're a student-led society based at the University of Oxford, which aims to bring talks by experts in academia and industry. Today, we have with us Professor William Adams. Bill is a geographer, and he's Emeritus Moran Chair for Conservation and Development at the University of Cambridge, and now also Claudio Segre Chair at the Graduate Institute for International and Developmental Studies in Geneva. He will talk a bit more about topics which he addresses in his recent book, which he co-authored with Kent Redford. So this book was released on July 27th by Yale University Press. So Bill will give a short talk, but then we'll have time for questions, and you'll be able to either ask them yourself by putting up your hand, or you can write them in the chat if you want me to read them out. So, well, thank you, Bill, for joining us today.
Okay, thank you very much. Thank you very much for having me. And it's very nice to be here. It's very nice for me as a geographer, as a social scientist, to be talking to people working directly in synthetic biology. So yes, the book that I have been involved in writing is called Strange Natures. And what I want to talk about today is the application of synthetic biology within conservation. And the book is co-written with a man called Kent Redford, who's an American ecologist. Back in 2013, I helped him run a conference in Cambridge on synthetic biology and conservation, trying to bring together people from engineering and genetics and biodiversity conservation and practitioners as well, to talk about whether there was a possibility that synthetic biology would have an impact on conservation, positive or otherwise. And we've just published this book, which pulls together some of our thinking on the subject. What I want to do today is simply ask or set out to answer 4 questions very briefly. The first is, why might conservationists consider genome editing and other applications to synthetic biology? Secondly, what are conservationists thinking of doing? What kinds of applications are being discussed in that conservation science literature? Thirdly, what questions are being asked and need to be asked about this use of genetic technologies? And lastly, will the public accept conservationists doing synthetic biology? So that's my structure, my four questions. So firstly, why genome editing for conservation? Well, the main reason for it is that nothing else that conservation does works. This is a slide from a new report on extinction rates, and you can see extinction rates rising for almost every taxon above the level which is in brown along the y-axis, which is the rate of extinction that would be expected naturally. Humans and our demands on the biosphere are driving species extinct, and that's what conservationists are trying to overcome. There are data on this, none of them entirely satisfactory, but this is one of the commonest indices used, the Global Living Planet Index from the Worldwide Fund for Nature. And basically it does what almost every other conservation graph does, and it goes down here from early 1970s to the middle of the last decade, a decline of 68% in biodiversity. And the main reasons for that are multiple: land use change, over-harvesting of wild species, invasive species and wildlife disease, pollution, and climate change. And basically the problem for conservationists is everything they do, the money they spend, the protected areas they acquire, none of it really makes any difference. And they look across at synthetic biology, they look at the rate of scientific advance, they see the applications in human medicine, in public health, in agriculture, in biofuels and biopharma, and they say, is it not possible that there are equivalent kinds of applications that we could use to tackle some of these threats, which other strategies don't seem to tackle? So that's why they do it. What are they thinking of doing? Well, 4 different categories I think one can think about. The first is the control of alien invasive species. The phrasing is rather strange, but basically these are species which occur outside their natural range because of human action. So people have taken species and either deliberately released them or accidentally released them. This one, for example, is a signal crayfish, which is an American species. In Britain, it has munched its way through all of the native crayfish, and it gets into rivers and it eats almost everything and burrows into the bank, so it destroys the fisheries. It's a real nuisance. There are lists of alien invasive species. The IUCN, the International Union for the Conservation of Nature, has a red list of threatened species. And in that, if you analyze that data, out of 17,000 alien invasive species, they're responsible for 126 extinctions. They're a factor in a third of animal extinctions and a quarter of plant extinctions. So control of alien invasive species is really difficult and really expensive. Here's an example of a problem. It's a very parochial problem for the UK. This is two species of squirrels. The one on the right is the squirrel that has long lived in the UK, the red squirrel. It's a native species. The one on the left is a grey squirrel, and it was introduced from the USA in the late 19th century. There are now about 2.5 million grey squirrels, and the red squirrel population has crashed to less than 200,000. And if you look at the maps on the right, you can see the declining areas of red squirrel distributions. Top map is 1945, the bottom map is 2010. The grey squirrel is gradually moving north across England, across Wales, into Scotland, into Northern Ireland, and indeed across Eire. It's not a direct competition. It's now known that the problem is partly that the grey squirrel has brought with it a disease, squirrel pox, and that hits the red squirrel very hard, so the population declines. And the conventional strategies are messy, expensive, and unpopular. They basically involve trapping or shooting grey squirrels. And they're pretty cute animals, and people quite like them. So it's not an easy kind of conservation strategy. Equally, if they don't control the grey squirrel, the other species will go extinct in the UK. So is there a strategy one could use using novel genetic technologies? Well, yes. A paper led by a team from the Roslin Institute in Edinburgh recently published a paper talking about the use of an engineered gene drive, which you probably know all about, but it's basically a device that will run through the population, and it will cut a target site in the coding sequence of the female fertility gene. And even though it's repaired, the female offspring are infertile. So basically, the recruitment of grey squirrels into the population would decline to zero. And population models show that under various scenarios, you would get local extinction of this species within 12 to 20 years. And in the paper, which is entirely based on modeling, they aren't doing this yet, they talk about this as an efficient, self-limiting, and controllable gene drive for managing invasive pests. And conservationists see us prick up at that, and they say, wow, this looks really neat. You do a hefty piece of science, and then you have something that doesn't require people with guns in local woodlands. It doesn't require poisons. It just lets this species die out. And they're thinking of the same thing in other contexts, for example, on oceanic islands where mice and rats are predators of endemic seabirds, very serious problem. Here up to the top picture on the right, an extraordinary and shocking picture. House mice have been released on Gough Island, which is in the South Atlantic, and they've grown so large that they actually attack the chicks of albatrosses and eat them in the nest. They kill them and eat them in the nest, and the albatross chicks just lack any ability to defend themselves. Or on South Georgia, here in the South Atlantic, a huge island, part of the British Overseas Territories, where rats had become established when there were big whaling bases there in the 19th and 20th century. And they just spent $11 million clearing South Georgia of rats, dropping poison bait from helicopters. They basically have to get poison bait onto every square meter of the island. And eventually the number of rats has declined to zero. That's costing about $100 per hectare. And people are saying, well, maybe there are cleverer, neater, lighter, less environmentally damaging solutions using genetic techniques. And this sort of application is really attractive to countries like, say, New Zealand, which has set a goal of a predator-free country by 2050. All the predators they're worried about are introduced predators, rats like the one sitting underneath the kiwi here, but also many other species. So there's a set of applications there which at least deserve conservation attention. Secondly, there's a set of applications to wildlife disease. There is, for example, a bacterial disease of amphibians, chytridiomycosis, which is responsible for the decline and extinction of quite large numbers of amphibians all across the world. And people are saying, well, maybe it will be possible, and researching whether it's possible to engineer the microbiome on the frog's skin to give it a resistance to these bacteria, and therefore to protect rare wild species against the attack of this bacterium. There's also talk of applying the same sorts of techniques to avian malaria. In Hawaii, there are large numbers of endemic bird species. There's one in the picture here, the scarlet honey creeper. And 25 of the 46 have gone extinct, and the others at a very perilous level. And one of their problems is they catch avian malaria and avian pox virus, which is spread by mosquitoes, both the disease and the mosquitoes introduced. And there's been a seminar discussing different sorts of control. The conventional method is insecticides and habitat reduction. So you take your strimmer and your insecticides to the environment. It's expensive, it's environmentally damaging, and it doesn't work terribly well. The other would be perhaps to use an engineered gene drive to eradicate mosquitoes by biasing the sex ratio of offspring, or to engineer the mosquito genome not to carry the pathogen. So they had this meeting back in 2016 talking about what needed to be done to protect the last of these endemic species. And among those were genetic techniques. The third field that's attracted attention is de-extinction, the recovery of extinct species using genetic engineering of the genome of living species, extant species. And National Geographic had a great splash on this back in 2013 with all these sorts of ice age animals walking out of the test tube onto a tundra. More realistically, the research that's going on, particularly led by the Long Now Foundation and the Revive and Restore program, is looking at this species, which is a passenger pigeon, which existed in huge numbers in North America prior to European annexation and settlement, and which went very rapidly extinct because it was hunted to extinction. And they're talking about editing the genome of a closely related species until they generate a bird which resembles the passenger pigeon, and then releasing that bird into the wild in the hope that it will start then to do the ecological functions of the species. So de-extinction is the one application of synthetic biology which really excites journalists. Bringing back the mammoth is a guaranteed article for the silly season in the middle of the summer to give your editor. And it's not the mammoth entirely unsuitable species, but it is possible that we will see people, we are already seeing people putting significant experimental money into manipulating the genomes of living wild species in the hope of creating something that resembles and has some attributes of something that's extinct. And the fourth area is what I call engineering adaptation to the Anthropocene. We're in this era where human impact is damaging the environment. So might it not be possible to use genetic techniques to undo some of that damage? For example, there is interest in engineering the genomes of corals to enable them to survive higher temperatures or greater acidity in the oceans. So saving the barrier reef using genetic methods. There are already corals, for example, those in Hawaii, which exist quite close to thermal vents in the ocean, and they cope with warmer temperatures than most corals can stand. And so, the idea, well, could you find out what the genes are that give them that capability and then insert those genes into other corals? There's also conversations about the engineering of organisms to detect or break down pollutants, or all sorts of applications to transform energy provision, biofuels and other sorts of things, or to produce something like synthetic meat directly, engineering products which would save wild habitat or improve the environment in some other ways. And that's the fourth thing. This is less directly the concern of biodiversity conservation. It's more of a broader sustainability application, but there's quite a bit of discussion about these things. And indeed, you can find quite a few papers that are talking about the possibilities of creating entire synthetic ecologies, not just altering domesticated species, but wild species and whole sets of species in ecosystems to create, for example, microbial communities, which are capable of coping with particular kinds of conditions. And people even use the phrase terraforming to talk about this. Sort of the idea that you could think about identifying the actual genetic diversity of the Earth, to redesign it to work better with the demands of 12 billion people. So the artist Daisy Ginsburg is very thought-provoking work she did a few years ago, where she talked about the standard sort of conservation, which works with past natural biodiversity, which is declining, and thinking about a future synthetic biodiversity with a mixture of synthesized genomes and naturally evolved genomes. So that's the wilder shores, you might say, of this kind of thinking. So what questions are there about this? Well, the thing that scares people in the conservation world is that if you start doing this kind of engineering to free mobile species in unbounded ecosystems, then there is the potential for genetic material to move out into wild relatives, to move ecologically with the organism if it escapes from the place where it's being managed into new context. So I put the European rabbit here, for example, a pest in many countries, a pest in the UK, to which it was introduced by the Normans in 1,000 years ago or so, a huge pest in Australia, a species of conservation concern in Spain. So if you start engineering rabbits, you need to be very sure where those engineered rabbits go. And the observation that edited genomes in wild species will evolve. You're putting something into the process of evolution, which you don't fully understand the consequences of. And the point really is about that the understanding of synthetic biology is mostly at the sub-organism level. It's how the genes work and how they're expressed in the organism. There isn't a great conversation between that and the ecosystem to understand how the ecology works, what would happen if you released a de-extincted passenger pigeon, or if you introduced 1,000 of them or 100,000 of them. What would happen? What would you expect to happen? How on earth would you know? Where are the models for that system? And it's that level of, that's the lack of connectivity, if you like, between the sophistication and the radical advances in genetic science and the relatively slow, limited understanding within ecology and conservation is the problem. And lastly, there's the problem about the proliferation of technology. If you can work out how to eradicate rats or mice on a tropical island, then why would you not find that technology owned by a pest control company in New York? And would it matter, therefore, if you lost this species from everywhere? And just what are those sort of consequences? That's not the animal getting out or the gene getting out. It's actually people saying, oh, that's useful, we can sell that, we'll have that. And that's what bothers people about it. And so everybody talks about the need for caution. Venki Ramakrishnan, the former president of the Royal Society, commented, if advances in genetic technologies have put us on the verge of a new age of biology, we need to go into it with our eyes open. The Economist back in 2018 talked about gene drives specifically, and I rather like their cartoon. Gene drives promise great gains and great dangers. Don't ban them. Don't ban and don't rush. It has to be done very carefully. So you have people looking at this technology from the outside and saying, okay, this is complicated, and the application of this needs enormously careful thought. But the technologies themselves are advancing incredibly fast because of people like you doing the research. The research frontier is enormously fast moving, and much of that research is done outside the not-for-profit sector, done in the profit sector, and therefore there's a vested interest in getting it done. And Faber and et al.'s paper on the CRISPR in the squirrels are very careful to point out that there is no legislative framework for the release of gene-drive organisms, and indeed it's currently illegal to keep grey squirrels in captivity, to even experiment on them. So we're a fair way from knowing how to make these things happen in the real world, and most of the papers are talking about what could be done, what might be done, and then arguing about whether if you did that, would it be desirable. And the thing that intrigues Kent, Redford, and myself most, I think, is what this power to edit genes, this power to create synthetic forms of life, does to our idea of nature. Because conservation basically is a social process. The conservation of nature is about people being persuaded that the products of evolution have value because they're not human. They're not art galleries. They're not great music. They're not human achievements. They're natural achievements. We think there's something there which is natural. And the question we ask in the book is, do genetic technologies disturb the boundaries between the natural and the artificial? Do they make arguments about loss and irreplaceability less convincing? In other words, do they undercut conservation arguments? And of course, do they offer new risks? And we point out that the decisions of conservationists on these points are important, but not really very important, because they're not the ones doing the science. So the fourth question is, will the public accept conservationists doing this? Well, there's a literature on that too. People like Ronald Sandler from Northeastern University in the US, talking about what needs to be done, offering an ethical framework for conservation projects that want to do this. A lot of work on risk assessment and management. His assumption is that this is all going to be done in a public forum, that the general public will have a role in assessing this. But somebody has to assess the goals, the means, and the desirability. Is the project justified? Is the goal justified? Can the goal of the project be accomplished responsibly? Is the project overall desirable? So think about bringing back the passenger pigeon. Is that worth it? Is it worth the effort? Can it be done responsibly? How many pigeons are going to die in the process? If this happens, are there people going to lose from farmers, for example, are going to lose with flocks of pigeons attacking their crops? And is it overall desirable? And he asks the question, which are the key questions, about who has oversight on these experiments? Who says whether they should take place? What's the role of the public in that oversight? What possibility exists for enforcement if somebody does something they shouldn't? And we saw a couple of years ago a Chinese research team editing the human genome in ways that were not judged ethical. How do you enforce? Who enforces it? If it's not against the law, how is it enforced? And what's the jurisdiction for this? If the UK works on the grey squirrel, which is in America, should American ecologists have a say in that, or is it every country for itself? Those are questions. Will we trust the science? Well, I think that's a very important and very tricky question. 3 or 4 years ago, the Royal Society did a big attitude survey on genetic technologies. And they found that attitudes depended on the use of the technology. So people were pretty happy when they'd been given information and asked to think about it, about the use of novel technologies to treat incurable life-threatening diseases in humans. They were pretty convinced about it for improving crops, for example, against fungal diseases. They were not at all happy with other applications like cosmetic changes to animals, micro pigs to sell as pets, or fluorescent fish to sell as pets. And they didn't think that making fruit and vegetables perfect in supermarkets was worthwhile. So the public is quite discerning about, if you like, what the purpose is, and it matters what the purpose is. It's not the technology. Will the public trust synthetic biology? They'll say, well, what's it for? What's the benefit for? And they're able to have quite a sophisticated conversation about that. Secondly, they found that trust in scientists depends on who employs them. Half of the people they spoke to trusted university researchers. Only 16% trusted scientists working within business organizations or funded by businesses. So who's doing the research matters to people. So trust is much more complicated than one might like to think, and it isn't going to come or in one go. It's going to be necessary to explain who's doing it and how and why. But it's happening. I saw this press release the other day from Rothamsted. Genome-edited wheat has its first field trial go ahead from the UK government. A number of people in synthetic biology in the UK are talking about the kind of Brexit dividend for research, because we are breaking away from the EU's very cautious position and moving to the much more free market libertarian position of the USA and other countries. So, for the UK, this is quite good news. And there are lots of applications out there. Agricultural pest control, work on the diamondback moth, which is a serious pest of brassicas, cabbages, public health, mosquito control, for example. Billions of genetically modified Aedes aegypti have been released in the Americas, and work on the American chestnut, which is a species that suffered from a fungal disease and died out in North America. And it's been back crossed with the Chinese chestnut, and now they've introduced a gene from wheat. So lots of examples sort of creeping into the world. And our book is saying, conservationists really need to get on top of this. They need to learn what these technologies do. They need to understand what they do, and they need to develop a fairly sophisticated discussion about what's happening. Because there is already a bad-tempered discussion about synthetic biology and genome editing, picking directly up from the GM crops debate of the 1990s. So you have some conservationists, like we should call it here, saying conservation biology needs a bigger toolbox, and part of that toolbox could be synthetic biology. And Smolker and Petterman, in their book Biotechnology for Forest Health, calling the genetically engineered American chestnut tree a Trojan horse because essentially they say the synthetic biology industry is funding this work in order to make people think that these are cuddly, easily controlled technologies. So there's the potential for a really bitter outcome out there. The issues in the conservation anyway are being discussed. After our Cambridge meeting, the Conventional Biological Diversity produced a technical report in 2015. The IUCN produced a technical assessment in 2018. And in the last few weeks, IUCN has been at its World Conservation Congress in Marseille, has been trying to discuss what its policy should be on synthetic biology. It isn't going to be easy because conservation and environmental NGOs and human rights and development NGOs intersect in quite complicated ways. This diagram is simply to remind me to talk about the web of concern. So convincing everybody that this technology works is not going to be particularly easy. There are lots of links across between organizations that are concerned about justice and poverty and peace, and those who are concerned about the environment. So the debate is on. That's the nice front cover of the IUCN report. This is a report that was published a few weeks ago, criticizing them, particularly for not calling out gene drives. And you have a press release on the right from an NGO saying that the synthetic biology industry, whatever they think that is, has hijacked IUCN. So the debate is starting. We've written the book to try and get it going. Our hope is that reading the book will help people to understand both what the technologies well, what the technologies are, and both what they can offer to, might offer to conservation, and also how conservationists might want to think very hard about certain kinds of applications. We've tried not to take sides in what is an emerging debate, but have written something I hope that people will find useful. In the process, it might be useful for people who work in synthetic biology, who actually want to scale up their thinking and understand how this might work in ecological systems. Right, I'll stop there. Sorry, I've gone a long time, Karina, but there we are.
All right, thank you very much for the talk, Bill. Yeah, we have time for questions now, so you can either use the raise hand function, and I will call on you to unmute yourself, or you can put your questions in the chat, and I can read them. So until we have some volunteers, what personally struck me immediately when I read the description of the book was that neither you nor Kent are biologists. I was surprised to see that actually this conference on synthetic biology and conservation happened in 2013. But do you remember like how it started, how you got introduced to synthetic biology?
Yeah, thanks. So Kent is a biologist. He specialized in ecology, and he then had a professional career in American universities and ended up as chief scientist for the Wildlife Conservation Society, which is one of the big 5 conservation NGOs. So I'm the odd person out. I'm a geographer and never trained to in biology. And it was Kent's idea to hold the conference in 2013. He felt that he'd obviously come across, he'd met synthetic biologists and had been inspired by the scope of the vision technology and the speed with which it was moving. And he persuaded me and Georgina Mace, who was a very eminent ecologist working in the University of London. He persuaded the 3 of us to work with him to host the conference. And his vision, I think, was a very simplistic one, which is if you put synthetic biologists and conservation biologists in the room together, you will spark amazing conversations. And actually, there were good conversations, but what I discovered is that most of the people who are doing synthetic biology gave up ecology as soon as they could because they didn't like it very much. So they gave it up in the first year. And most of the people doing conservation gave up genetics as soon as they could and gave it up in their first year. So actually, they didn't know enough about one another's science. They made assumptions about how ecosystems work because they understood something about how genetics worked, or the other way around. And that actually the work, the communication difficulty, the interdisciplinary communication difficulty, even within people who I thought of as biologists, within that discipline is a myriad of disciplines, and that perhaps we've all become very siloed in the way we learn about things, and so we just don't have much expertise outside our area, and that matters here. Because the expertise of one area has enormous potential, good and bad, for another. And the public expects scientists to communicate, but actually we're not very good at communicating.
Thank you for that. I should have been more specific on like the molecular side of biology. So we have a question from Jesse. Would you like to unmute yourself?
Yeah, thanks, Karina. Thanks, Bill. Yeah, so I'm just here's a question. So you're a geographer, and I've also kind of heard other geographers talking about synthetic biology, and that's someone named Tim Kruger here. So that but it comes from more of an ethical perspective, I think, and thinking maybe about some of the implications. And I'm kind of interested. Okay, well, are there people that work on, do you know of any people that actually work on the interface between synthetic biology and, say, geosciences or, geography in that case, with kind of developing some of these techniques? Because that's ultimately what I'm interested in, but I find I struggle to kind of find find kind of examples of where that's been done already. And maybe it hasn't been done.
Yes, I mean, I've seen articles written about it, but one of the troubles with researching across a wide front on this is that an awful lot of things are written which are essentially thought experiments, often by companies with new products or with potential new products, or scientists, at conferences trying to sort of do the thinking around the edge. So a lot of it's quite speculative, and there's a big, Kent keeps reminding me, there's a huge distance between a paper saying, hey, we could do this, and actually a proposal being in a state to go to some assessment agency. So yes, I've seen people writing about the role of large-scale biological systems, ecosystems within atmospheric gas management. So the whole idea of planetary engineering sort of has a very strong biological component, and therefore that field needs to move a long way beyond sort of dropping iron filings in the ocean in the hope that there'll be a plankton bloom, to actually looking at the biology of the plankton. And of course the biology of the seabirds and the whales and the fish and etc. So frankly, I think that the climate change community isn't very comfortable with biology. I think there's a lot of large-scale or small-scale biology which that isn't in their training, because they're often coming at it as trained as physical modelers of physical systems. So there's a lot of communication to be done, and I don't know which research groups are doing it, but I do think that the planetary engineering community need to get good at biology, both sort of molecular biology, but also ecology, pretty fast, because I say the public expects joined up thinking, and
That's right.
we spend 150 years not talking to each other, creating disciplines. We train people in disciplines, and then somehow we expect there to be a flowering of communication. And it's really hard if you try and listen. I mean, I feel this, listening into reading the technical, trying to read the technical literature on synthetic biology. It's really tough. And if you tried to write it in the way that would make sure that no ordinary person would understand it, that's how you write it. With exceptions, of course, there's lots of good writing, and Nature and Science and other journals like that make the effort. But there is a real challenge here.
Yeah, exactly. Yeah, well, I'm in the Earth Science department, so I'm here to learn and to talk to synthetic biologists. So drop me a line if you want to chat. I'm here in Oxford.
Good luck.
All right, thank you. So we have a question in the chat from Nathan, who thanks you for the talk, Bill. And he's interested in synthetic biology, the gene drives for public health issues, stating that he thinks the mutation rates of some of the disease-causing, like viruses or parasites, would be quite difficult to overcome. Can you comment on that?
Yeah, look, I'm not. I would intuitively imagine that that's correct, but I just don't know. That the way they're focusing on these things is on the vectors, on the mosquitoes, and they're really just looking for a way of knocking populations back. So there really isn't an expectation, for example. So, for example, a lot of work is going into malaria in Africa, which is still a major killer. Kills mostly children, but lots of people dying of malaria. So getting rid of that would be a real sort of humanitarian success. And realistically, they're not imagining to drive the mosquitoes extinct. They just want to get the populations right down. Now, at the moment, if you want to do that, you have to use pesticides, and they're pretty nasty things. And you're killing lots of non-target organisms. Some of the pesticides are actually toxic, directly toxic to humans. So there's not a good kind of strategy. So the notion that you could hit populations using a genetic technique is attractive, but they'll come back, they'll reinvade, and they'll come back. Indeed, from an ecological point of view, since lots of lots of birds, some of them rare, other insects, they eat insects. I mean, mosquitoes are pretty important sort of food source. So, but yeah, I'm sure you're right about the disease, but I'm absolutely not enough of a scientist to know how to answer your question.
That's okay. So Jasmine is also asking a question in the chat, and I'm going to just read it out. Are you aware of ongoing discussions about digital sequence information within frameworks relating to the Convention on Biological Diversity? And do you think what you've discussed here has implications for access and benefits sharing within the CBD?
Thank you, Jasmine. Yeah, that's a really good question. Yes, it does. And I mentioned that the Convention on Biological Diversity had done a report and has asked one of their technical subcommittee to look at synthetic biology. And they seem to me to be pretty much stalled at the moment, partly because it does undercut a lot of the principles on which they were based. So this is a convention which was agreed in the early 1990s. And one of its provisions was an attempt to protect indigenous knowledge, indigenous varieties of medicines or crops, so that the benefits should not be simply captured by the person who took the specimen, but should flow back to the people who had bred the species or had the but of course, you don't need the specimen anymore. You just need to know what the genetic sequence is. It does change the dynamic of the flow of materials that you can use for creating biological products. And it makes much more complicated the question of of access and benefit sharing, who ought to have some licensing benefit. And the kind of notion was, if you have a European or an American pharmaceutical company, and they send somebody out into the Amazon rainforest to meet someone who lives there and who's a local healer, who says, oh, when someone has a disease, we always give them this root, this plant root. And the guy says, oh, thanks very much, takes it back, and then turns it into a medicine and sells it for millions of pounds. Surely some of that money should go back to the person, to the community that had the knowledge. That's the idea of it. And yes, it does change all of that. And I think people are just trying to start to work out how to think about it, what it means. I mean, there's all sorts of things you can do. I mean, you could you could quite easily synthesize, well, you can synthesize real vanilla. It's not imitation, it's actually vanilla. And so what does that mean if you're a vanilla farmer in Madagascar? They're among the poorest farmers in the world at the moment. They have a product that we like to use in our ice cream and so on. But what if there is no difference between a synthesized vanilla and that? Or what if you could make a vanilla plant that would grow in Europe or North America? You wouldn't need to import it anymore, which is great for us. We have lots of cheap vanilla. It's not so great for the farmers. And that consequence ripples down. No one's really thought about that or modeled it in lots of ways. So yeah, really good question. Thank you.
Thank you for that. So Shyama from the Wildlife Conservation Society in India had a question. Will climate change conversations on nature-based solutions provide space for synthetic biology, or are the solutions pointing towards a different direction?
Thank you, Shyama. That's a good question. So nature-based solutions, is an argument that conservation is using at the moment, which is that natural habitat, wood forests in particular, but also other habitats, are really significant in ways such as storing carbon. And so protecting forest and other sort of areas of natural ecosystems is a really important part of the response to climate change. So alongside all of the moves we need to make to a less energy guzzling economy, we also need to protect nature, and it's really important. So Shyama, I think at the moment conservation is mostly using that argument about to promote the protection of the pristine. They're mostly using it to say we have to preserve this species because it's valuable for its timber and for its carbon. I think conservation is a bit reluctant to say, well, actually, we should take the forest away and plant something different, which would store more carbon. We say, no, that's not what we meant. But as it were, the notion that you might say, well, we could take these trees or we could take these plants and we could actually use genetic technologies to increase the amount of carbon they sequester. We could release bacteria into the soil of this nature reserve, which would lock up more carbon. And those kind of applications, I think, are not what conservationists are thinking about in terms of nature-based solutions, but they are things that people will start to think of doing. And my point is simply that we need to understand the ecological and indeed the social and economic consequences of those kind of things, rather than just saying, yeah, that would work. That was a cool idea. Yeah, you could do that. No, absolutely, you could do that. And say, yeah, but what happens if you do that? And I think I have to say, I think that's not within the skill set of the synthetic biology community. They need to bring on board other people who can understand those kind of consequences. And it asks questions of ecologists that are absolutely not able to answer. We don't know how ecosystems work very well. We have far more knowledge of genetics than we do of ecology. And there's far more money going into research on it. So, I think it's going to be a tricky conversation. But yeah, really interesting that you should make that link. And it's a really important thing to think about. Yeah.
Thank you. Nathan expanded on his question, I guess asking whether introducing these pressures of gene drives, it could be possible that some mosquitoes that survive would come back and wreak havoc, that we've somehow selected for something more detrimental than what we had before. Is that a concern? Or any way feasible?
Yeah, absolutely. So, I mean, I think that thought experiment is really important, and one needs to understand the dynamics of these genetically novel organisms within an ecological context, accepting that ecological context is also very often a human influenced ecological context, and that therefore, understanding their impact is really, really important. So yes, you don't know fully what the implications in terms of fitness, in terms of interactions between organisms of a change are. And so where they do, scientifically, that needs something we really need to study. And that is why members of the general public are often quite scared of this stuff, because they assume that something terrible might happen. And of course, if you know the science well, you think, well, it's very unlikely to happen. And they say, yeah, how very unlikely is it to happen? And they think of all the other things that other scientists have said wouldn't happen and did happen, and they link them together and they say, well, this is really scary. I don't like this at all. I don't think you should do this. And that's pretty much where the debate is at the moment. So yes, I think there are questions about, the scaling up from the genome to the ecosystem is really challenging scientifically. And that cross-scale thing is really, really important. That study is really important. And at the moment, it's not really getting a lot of research funding. There's a lot of research funding to look at the genetic level, to think about the technologies. There's much less to look at the wider questions, let alone the ethics and other issues. So there's lots to do, I think. And it is that fear, Nathan, it is that fear of unfavorable outcomes, that the possibility and how you do the science to be able to say, how likely that possibility is and how consequential it would be. And then how you equip the public to accept the results of that careful science. I mean, we've seen a lot happening around. If you think, I mean, Kent and I wrote a piece the other day about vaccines, and you can see. I mean, we've seen in the UK, for example, a lot of knowledge now about viruses and vaccines, which no one had before. People understand infection in a way they didn't. they understand a disease transmission in the way they didn't. But it's not that everybody says, oh, thank you, science, we really understand that. There's also all sorts of layers of worries and concerns in the vaccine hesitancy, which has risen and so on. So that's just one area. We need to be able to do that with other things and not move so fast that we expect some senior scientist in front of a microphone saying, oh, it's okay, we've done the science, you've got nothing to worry about, it'll be fine. People aren't minded to accept that. they think, oh, that just sounds like some kind of cover up to me. And you think, well, hang on, I'm a scientist, I don't cover anything up, that's not what I do. And they say, well, I don't believe you. And that's the challenge we've got.
Thanks. I think Sarah is also asking a very important question, like, would it not be better to find ways to restrict or decrease the human changes that cause such events such as coral bleaching instead of genetically engineering the coral to make them more heat resistant? So are we using synthetic biology to change things so they're good for today's world when really we should be changing the world and the impact we have on it?
Yes, Sarah, that's a really good question. Yes, I think I agree with you. It's been interesting writing this book with Kent Redford because he's probably more engaged with the positive possibilities for conservation synthetic biology, and I'm probably more engaged with the possible negativities. So there is an argument that says, biodiversity loss is acute and very rapid, and we have to do everything as quickly as we can to save biodiversity. And there's another argument that says if you don't address, and that leads you to creating large areas of national parks and throwing people out of national parks to protect nature, all those kind of things. There's another argument that says unless you address the fundamental drivers, which are economic growth, the assumption that economic growth is always a good thing, the assumption that economic growth will always trickle down, the nature of energy demand, the nature of material demand in our production and consumption, the way we buy clothes, the way we buy, unless you address that, you're never going to win. And simply jumping to a technological solution isn't the right solution. So it's rather like the argument about carbon burial, for example. There are people who say we should do this. We should find a way of locking carbon up in rock, and that's how we should address climate change. And among the people who are funding that are all the oil companies that would like us to keep using oil. And so similarly, people are saying, well, synthetic biology is actually just it's just attractive to all those powerful interests that like the way the world works, they profit from the way the world works, and that maybe we need to actually address our own consumption demands more directly. So you put your finger on a great challenge there for us at the moment.
Okay, I think we should be wrapping up soon, so I'd have one more question for you, Bill. Looking up your CV, basically, I found that you already wrote a book called Future Nature, A Vision for Conservation in 2003. So that was when synthetic biology was very much in its infancy, if it's been out at all. So how have your views changed since then? Like, did you see genetic technologies playing such a role?
That's a great question. Look, in 2003, I had never heard of synthetic biology, and my knowledge of genetics was that of a schoolboy. So I think I've learned a heck of a lot from the way it happens. What I found interesting is that in a sense, if you read the popular science literature, the sort of websites and so on about this, a lot of the articles have a particular kind of form. They are sort of huge problem needs to be addressed very, very rapidly. Radically new science. It'll all be brilliant. And it's sort of it's all in sort of capital letters and bright colors. And I think that we're stuck in this mode with sort of synthetic biology, you sort of pulled out as a solution to whatever problem you might have. And I think we have to get through that until we start to see things that it does really well as a technique. I mean, for example, it's really useful, it turns out, if you're making vaccines very, very fast. You know, and that's a really clever thing. And other places where maybe one wants to be very, very cautious about large-scale applications. The book on future nature was really a challenge to, actually, it was very much focused on the UK, and it was a challenge to think about nature not just as beautiful wild things in wild places, but to think about nature across the board, to think about nature in cities, to understand that humans and other species live together in quite complex ways, and to try and think about a conservation future which gave you beauty, natural beauty and diversity in and around people, not simply over there on a David Attenborough documentary or in a nature reserve that you can only visit once a year. And in a sense, I think by thinking synthetic biology fits into the same kind of vision. It seems to me we need to be holistic about it. We need to understand that we're actually talking about the biological system of which we're part. And we need to think quite carefully about our relationships with other species and with the technologies that we use to interface with them. And you know, synthetic biology offers us a radically new, potentially hugely powerful set of technologies, and we need to understand them as fully as we can. We need a conversation that's as diverse as it can be about what they're good for and what we shouldn't use them for.
Yeah, that's an important conversation to have. So thank you very much, Bill, for giving this talk today and for answering all these questions. And thank you to all of you for joining us today. This talk is recorded and we will soon put it on our YouTube channel, and you'll certainly hear more from us as we're slowly coming out of our summer break. But for now, this is it. Thank you, everyone. I hope you have a nice rest of the day. And if you're in the UK, please go enjoy the sun now.
Okay, thank you very much, everybody. Thanks for having me, Corinna.
Bye, everyone.